Myself, Dr. Vasantarao B. Kadam, Professor, Department of Botany, KTHM College, Nasik. I am a research guide for PhD and MPhil students. Under my guidance, four students are completed PhD and three students are completed MPhil program. Now presently, five students are working for PhD. Out of five, two students are under environmental science and three students under botany. I have published 150 research paper in various national and international journals having good impact factors. I am the member of editorial board of 25 various national and international journals, written 10 books for UG, PG and research students. Students, today we are going to learn the topic plant tissue culture, bamboo tissue culture under the STRIDE program. The complete bamboo tissue culture is divided into three sections. That means our today's lecture is classified into three various parts. One is structure of tissue culture laboratory. That means the basic structure of tissue culture laboratory. Second, micropropagation of bamboo. That means the journey of preparation of plant tissue culture bamboo from selection of suckers to the hardening and actual plantation of bamboo in field. And third part, content advantages of bamboo tissue culture plants. What are the benefits? What are the advantages of plant tissue cultures? So students, we are going to cover the three parts of our today's lecture. First, structure of tissue culture laboratories. Before we start our structure of tissue culture laboratories, we know the basic definition of tissue culture. Tissue culture means culture of any vegetative parts of a plant in nutrient media under aseptic condition for a regeneration of identically mother plants. New plants are regenerated through this process is known as tissue culture. The structure of tissue culture laboratories are classified into two various zones. First, sterile zone, and second zone is known as non-sterile zone. Under sterile zone, having the various compartments. First, media storeroom, second, culture room, growth room, and change room are complete sterile atmosphere maintained in various four compartments. And in non-sterile zone, washing places, chemical store, and media preparation are the three compartments, are not the sterile compartments, are not maintaining the sterility, but maximum hygiene condition is maintained throughout the day in non-sterile zone. Non-sterile zone having first chemical store. Various chemicals required for tissue culture laboratories for plantation of tissue culture plants are stored in according to its temperature, storage temperature, and it contains agar-agar as a gelatin agent, carbon source as a sucrose, microelements, potassium nitrate, ammonium nitrate, Microelements manganese, molybdenum, zinc, boron, copper, and iron are used for the tissue culture nutrient media. Number of vitamins, mm, myoinositol, thiamine, pyridoxine, and nicotinic acid are also used. Amino acids are also used in the nutrient media. Glycine, arginine, asparagine, aspartic acid, alanine, glutamic acid, glutamine, and proline. Basically, 20 different types of amino acids are synthesized in plants, but some amino acids are supplementary used in nutrient media. Plant growth regulators, auxin, cytokinin, gibberellin, abscisic acid, and ethylene are the various growth hormones are used according to the requirement of the tissue culture conditions. For example, cell division, root formation, shoot formation, elongation. The required hormones are used for the preparation of nutrient media. Second, media preparation room. In media preparation room, the various kinds of glasswares and instruments are used for tissue culture and are different types of glasswares, different kinds of balances, required chemicals, hot plates and stirrer, water bath, pH meter, autoclave and hot air oven, microwave oven, vortex second, centrifuge, refrigerator and freezer, storage cabinet, etc. Ingredient for media preparation: carbon source. The most commonly used carbon source is sucrose. It is readily assimilated and relatively stable. Other carbohydrates like glucose, maltose, galactose, and sorbitol can also be used and may prove better than sucrose in specialized circumstances. We used sucrose as a supplementary food because the inoculated plant or inoculated explants are not doing photosynthesis. That means they are unable to do the photosynthesis, means preparation of its whole food material. That's why we used sucrose in nutrient media as a supplementary food material for the cell division of the explant. Gelatin agents, plant tissue culture media can be used in either liquid or solid form depending on the type of culture being grown. Agar produced from seaweed, it is the most common type of gelatin agent and is ideal for routine application. The agar agar is made up from the red alga, the member of Rhodophyceae. The main industries of agar agar is based at the Goa. In Goa Sea, the red algae seaweed is collected and prepared the agar agar by the various agar agar industries. Agar agar is used for the solidification of media. 8 gram of agar agar per liter of water, distilled water, are used. If we need the semi-solid media, then we reduce the content of agar agar. That means 5, 6 or 7 gram of agar agar in 1 liter of distilled water. The main function of agar agar is the only solidification of media, and sucrose we used 30 gram per liter of distilled water as a ready carbon source. Number of microelements, macroelements are also used. Potassium nitrate, ammonium nitrate provides nitrogen source to this media. This mixture of cation and anion balances the pH of the medium. Also, this ratio play a very important role in plant growth. Microelements manganese, molybdenum, zinc, boron, copper, and iron play an important role in plant metabolism, while manganese plays a vital catalytic role in nitrogen metabolism. Micro elements and macro elements are already present in soil. It dissolve into water and through roots they absorb by the roots of plants. Now, plants having the macro and micro elements already absorbed by the roots, which is dissolved in soil by active and passive absorption of water. And for tissue culture, the culture of explant also required the micro elements and macro elements for the better growth and multiplication, cell division. So we supplementary use these macro elements and micro elements into the nutrient media. Vitamins myoinositol concentration was increased as it shows si- stimulatory effect in callus. Increased concentration of thiamine, pyridoxine, and nicotinic acid have a stimulatory effect. Thiamine in the medium is a key element of carbohydrates metabolism and biosynthesis of some amino acids. Ascorbic acid is provided to control phenol production in the medium. So vitamins are also required for the better growth of the plant's material. So we provide a number of vitamins in nutrient media at the time of preparation of media. Amino acids, the most commonly used amino acids is glycine. However, arginine, asparagine, aspartic acid, alanine, glutamic acid, glutamine, and proline are also used. Amino acids provide a source of reduced nitrogen and, like ammonium ions, uptake causes acidification of the medium. Casein hydrolysate can be used as a source of mixture of amino acids. Students, 20 different types of amino acids are present in plants, are synthesized in between plants. So we use the supplementary amino acids at the time of preparation of media for the better growth of the plant material. Plant growth regulators. There are four main classes of plant growth regulators used in plant cell culture and are namely auxin. Auxin promote both cell division and cell growth. Indole acetic acid or indole 3 acetic acid is the most important naturally occurring auxin, but its use in plant tissue culture media is limited because it is unstable to both heat and light. 2,4-dichlorophenoxyacetic acid, that means T4D, 2,4-D is the most commonly used auxin and is extremely effective in most circumstances. Number two, cytokinins. Cytokinins promotes cell division of the naturally occurring cytokinin. Only zeatin and 2IP, 2-isopentyladenine have some use in plant tissue culture media. The synthetic analog kinetin and BAP, benzene aminopurine, are used more frequently in tissue culture. media. Number three, gibberellins. Gibberellins are involved in regulating cell elongation, in determining plant height and fruit set. Only a few of these gibberellins like GA3 are used in plant tissue culture media. And number fourth, abscisic acid. It is used in plant tissue culture to promote distinct developmental pathways such as somatic embryogenesis. Abscisic acid inhibits cell division. Number three, non-sterile zone is washing places. It is very important for a tissue culture laboratory. It should be provided with a large sink, running hot and cold tap water, various types of brushes, detergent, and buckets of single distilled water for a final rinse of the washed glass goods. A number of plastic buckets are required for soaking the glass goods to be washed. Another separate buckets with a lid is also required for disposing of the used or infected media before cleaning. Only this bucket should be kept outside the room or cleaning area and should be cleaned twice in a week. Now, the next zone is sterile zone where the complete sterility is maintained. This sterile zone of media store is highly sterilized. It is 2 to 3 times more sterile than our operation theater. First, media storeroom. Sterilized media stored in the media storeroom. Sterilized media used after 72 hours. After sterilization of media, it is stored into media storeroom and it is not used before 72 hours for production because if the media is not completely sterilized, then number of bacterial, algal, or viral infection shows before 72 hours. A continuous screening under the light is occurs in media storeroom before the media used for inoculation. If the media is completely sterilized, no infection in the media bottles, then the media is used for the tissue culture purposes. That means after 72 hours, if the infection is not shown in the media bottles, then the media is completely sterilized and it is ready to utilize for the tissue culture purposes. A regular screening work is done for observation of contamination and contaminated media instantly removed, discarded from the media store. Next, culture room. The laminar airflow cabinet is the most common accessory used for aseptic manipulation nowadays. The cabinet may be designed with horizontal airflow or vertical airflow, where the air is forced into the cabinet through a bacterial HEPA high efficiency particulate air filters. Through this high efficiency particulate air filter, 2 to 3 micron particles are filtered. Complete sterilized air is coming on the laminar airflow cabinet. The airflow over the working bench at the constant rate which prevent the particles, that means microorganisms, from setting on bench. Before operating in the laminar airflow cabinet, the interior of the cabinet is sterilized with the ultraviolet germicidal light and wiping the floor of cabinet with 70% alcohol. Before starting the actual work, the workers are first start the ultraviolet light which is fixed in the cabinet, laminar airflow cabinet. For 15 to 20 minutes, the ultraviolet light is continuously on. That means the ultraviolet light kills the inner side of laminar airflow cabinet. A number of bacterial viral infections are completely killed through this ultraviolet light. And the lower portion of cabinet where the actual cutting work is going on, which is cleaned by 70% alcohol. Why we use 70% alcohol? Because all known microorganisms are completely killed in 70% alcohol. That's why we used 70% alcohol. If we use 80, 90 or 100%, that means absolute alcohol, it is also okay. There is no problem. But it is all viral, bacterial, algal infection are killed in 70% alcohol. So we used 70% alcohol for cleaning the cabinet. Laminar airflow has a number of small blower motors to blow air, which passes through a number of high efficiency particulate air filters. Such filters remove particles larger than 0.3.2 micron. The ultra clean air, which is free from fungal and bacterial contaminant, flow at the velocity of about 27 plus or minus 3 meters per minute through the working area. All contaminants are blown away by the ultra clean air, and thereby an aseptic environment is maintained over the working area. Before starting work, laminar airflow is put on for 10 to 15 minutes. Number 3, growth room. The growth room means the room for keeping or incubating the culture under controlled temperature, light, and humidity. To maintain the temperature around 25 plus minus 2 degrees centigrade, insides of culture room, air conditioners are used. This room is also provided with specially designed cells to keep culture vessels. The cells are made of glass or polywood. Flasks, bottles, jars, petri plates can be placed directly on the cells. Cultures can be grown in light or in dark. Students, in growth room, after culturing the or after inoculation the plant material in culture room in front of laminar airflow cabinet, all culture bottles are placed in growth room for 1 month. 50 ml of nutrient media is provided in bottle, and it is sufficient for the 4 week or 1 month of the growth of these cultures or explants. In growth room, 24 hours the temperature is maintained 25 plus minus 2 degrees centigrades. Humidity is also maintained by humidifier, and continuous screening by the workers occurs in growth rooms. And the light intensity of light is 3000 lux. The light intensity is also measured 3 times in a 24 hours. If the capacity of light is low, that means it is less than 3000 lux, then the light is replaced by electrician. And in daytime, continuous screening occurs by the workers. If the contaminated bottles seen by the workers, they removed from the cells and discard. This type of work is continuously going on in growth room. Next is change room. Workers are wearing sterilized clothes, masks, caps, and aprons before entering in the non-sterile area. The workers are enter into the laboratory, the main door having the air blowers. Workers are standing for 1 to 2 minutes under the air blowers. The dust particles are present in our clothes, hairs, and our body parts are completely removed, and then workers enter into the actual tissue culture lab. First, workers going to washing area. They wash hands and legs. Then next, entering into the change room. In change room, according to the number of workers, the apron, cap, and mask are completely washed and sterilized. Packets are present. The workers are taking our mask, cap, and aprons. After wearing the mask, cap, and aprons, the workers are actually entering into the tissue culture lab where they are working in storeroom, culture room, or in growth room. The three rooms are completely sterilized. Students, we learn the basic structure of tissue culture laboratories. This is the first section. We finished here. And now the second part of our presentation is micropropagation of bamboo. That means selection of explants to the production of tissue culture bamboo plants. The complete journey we are going to learn now in second section, and which is micropropagation of bamboo. Students, we are going to learn the second part is known as micropropagation of bamboo tissue culture. The process consists of 8 important steps. And first is selection of explants. Number 2, virus indexing, surface sterilization of explants, initiation of explant, multiplication, shooting and rooting, primary hardening in greenhouses, and secondary hardening in shade houses. First, selection of explant is the most important criteria for the tissue culture laboratories because we took the explant from the mother plants and we prepared number of plants through tissue culture, and all plants are identical to the mother plants. That means the selection of mother plants for taking suckers is the most and basic important criteria for each and every tissue culture laboratories. Seeds, seedlings, inflorescence, root culm, mature clumps, nodal segments, meristem domes, or leaves are used for bamboo tissue culture. First criteria for selection of explant mother plants are height of mother plants, number of leaves, leaf area, and color of leaves. If the number of leaves is more, leaf area is more, and color is very good in grain, so the rate of photosynthesis is more. That means preparation of food material is more. Naturally, the production of plant is good. And next is orientation of plants. The height of plants and orientation of plants are the most important criteria for the selection of mother plants. After selection of mother plants, we took the explants, a plant material from the mother plants, and in tissue culture laboratories, a small laboratories is also working with the help of the tissue culture laboratory, and it is known as virus indexing laboratories. In virus indexing laboratories, the selected explants are transported from field to virus indexing laboratories for testing the viruses in explants. If the explant having the infection of various kinds of viruses or diseases, the explants are discarded. It is not sending towards the tissue culture laboratories for multiplication. The explants completely free of viruses, free of diseases are used for the tissue culture laboratories and are transported from virus indexing laboratory to tissue culture laboratories for actual production of tissue culture plants. So the virus indexing laboratories is the most parts for identification of disease-free, virus-free explants. On bamboo plants, some viruses are basically present and are bamboo mosaic virus. Is a plant pathogenic virus in the genus Vortexvirus and the family Alphafexiviridae. The bamboo mosaic virus is a filamentous flexus rod 490 nanometer in length and 15 nanometer in width. The virus has been fully sequenced and it is 6366 nucleotides. That means 6366 nucleotides long. Too much long. It was first isolated in 1974 from two species of bamboo and are Bambusa multiples and Bambusa vulgaris in Brazil. The picture shows the infection of bamboo mosaic virus on leaf for identification of the viruses. Number two is cherry necrotic rusty mottle virus. Cherry necrotic rusty mottle virus has mainly been associated with disease of sweet cherries. The first incidence of the disease was described in 1945 in Utah, and since then the virus has been reported from many cherry growing regions of the world like China, Japan, Europe, Germany, India, Korea, New Zealand, and North America. The virus is also present on the bamboo plants. The leaf sheaths covering the nodal segments containing axillary buds 1.5 to 2 centimeter were carefully removed and wiped with 70% alcohol, ethanol using sterilized cotton. The explant were surface sterilized in 0.1% volume by volume solution of mercury chloride for 15 minutes and washed 4 times with each washing step lasting 5 minutes. After the virus indexing, the explant is completely disease free and now these explants are surface sterilized by these various processes. After surface sterilization, the explants are transported into the culture room where the workers are sitting in front of laminar airflow cabinet. And now the use of this cabinet is to maintain the sterility or maintain the sterile atmosphere during the culturing of these explants onto nutrient media bottles, and it is known as initiation. After surface sterilization, the plant materials are initiated. The explants from mother nursery are sterilized. Aseptically dissected shoot tips are inoculated onto specially formulated nutrient medium for proliferation of shoot buds under controlled clean room conditions. And now the workers are putting these explants into freshly prepared nutrient media bottles, and this process is known as initiation of explant. This is known as zero generation in tissue culture laboratories. And after inoculation, all inoculated bottles are transported from culture room to growth room, where the bottles are placed for 4 weeks, that means 1 month. And after 1 month, the explants are multiplied. Next step is shoot multiplication. Proliferated axillary shoots of 3 to 5 shoot clusters were excised and transferred to MS medium supplementary either with BAP at 5 levels. 5 levels means various kinds of concentration: 1 milligram per liter, 2 milligram, 3 milligram, 4 and 5 milligram per liter are the various concentrations. 6 milligram per liter kinetin are supplemented in nutrient media, or combination of kinetin at levels 1, 2 and 4 milligram per liter with optimum concentration of 3 milligram per liter of BAP. It depends on various species. It is worth maintaining that the choice of 6 milligram per liter of kinetin at one level was based on preliminary finding where best effect was observed. Shoot multiplication was performed using 3 to 5 axillary proliferated shoots excised from the clusters of shoots. Multiplies, Multiplied shoots in liquid cultures were supported by sterile filter paper bridges. Regularly, the shoots were transferred at intervals of 10 days under sterile condition to fresh medium in order to prevent phenolic oxidation that can trigger the shoot to become yellowish. The multiplied shoots on each culture medium were counted after 45 days to evaluate the multiplication rate of the nodal explants. This picture shows the multiplication of explants having roots and shoots. The workers are observing the growth, roots, and shoot height, and also they observe the contamination or the various media bottles having the multiplied shoots of bamboo. Next step is shooting and routing. Multiplied shoots are individually separated and graded for quality. Individual shoots are then transferred to another nutrient medium for shoot elongation and root development. Now, in nutrient media, some hormones are used for the development of root and shoots. Fully developed plantlets are sent for primary hardening in specially designed greenhouses. Rooting process. We used 5 levels of sucrose in half strength of MS media to determine the optimum concentration that induces rooting. Furthermore, rooting was achieved by specially transferring multiplied shoots to half strength MS medium. Morasis and Skook are the scientists that developed this media, prepared with 3% sucrose and supplemented with different combination of growth regulators as follows. We used 3 milligram per liter of indole 3-butyric acid, 3 milligram per liter of IBA with 10 milligram per liter of coumarin and 3 milligram per liter of IBA, 3 milligram per liter of indole 3 acetic acid and 10 milligram per liter of coumarin. All cultures in the experiment were incubated in growth chamber at 25 degrees centigrade with a 16-hour photo period at 30 lux light intensity. Out of 24 hours, 16 hours provided the light and 8 hours darkness. That means the cycle of light and darkness is required for the photoperiodism. Routing percentage and average number of days for routing were recorded. The tissue culture plants are grown under aseptic condition, under controlled climatic condition. That means 25 plus minus 2 degrees centigrade. And now the plants are hardened and kept into the room temperature. And the room temperature is 27-28 degrees centigrade. The room temperature for 4 to 5 hours, the plants are placed into without AC room. That means they are slightly hardened or acclimatized to survive the normal temperature. The rooted plantlets are kept at room temperature for certain time. Then they are taken for acclimatization process where they gradually tend to the in vitro temperature and light. There are two stages in hardening process: primary hardening in greenhouses and secondary hardening in shade houses. After 4 to 5 hours, the bottles are placed into room temperature without AC and then transported towards the primary hardening into greenhouses. Now, the primary hardening occurs in greenhouses where the temperature is controlled up to 29 to 30 degrees centigrade. Optimum microclimatic conditions, relative humidity, air circulation, light intensity, and temperature are maintained. In vitro produced plantlets are removed from the glass jars, washed and gradually and graded for quality. Now each and every bottles are removed the plantlets, wash the roots because roots are having the media. After washing the plantlets are graded into according to height: A grade, B grade, and C grade. Larger plants are under A grade, medium plants are under B grade, and lower plants means less heighted plants are under C grade. After separation of grading, each graded plants, for example, A grade, B grade, and C grade, are planted into potrays, plastic trays, having wet mix, soilless media. The A grade potrays, B grade, and C grade separate planted according to the height. These plantlets are then hardened under controlled microclimatic condition in specially designed polytunnels on benches. Greenhouses help in bringing out healthy primary hardened plantlets. Now, the irrigation and fertigation of the plantlets are given by sprinkler irrigation system. Highly stringent hygienic condition as well as controlled microclimatic condition in the primary hardened facility are religiously maintained for achieving quality of plantlets. Now, in 1.5 months, that means 45 days, the plants are placed in greenhouses. After 1 feet of plants are transported for secondary hardening into shade houses from greenhouses. And now the plants are primary hardened. They are survive for in atmosphere around 29 to 30 degrees centigrade. This is the picture of separation of the gradation of plants A grade, B grade, and C grade by workers. Now, this side picture having after removing of the plantlets into bottles, washed, see the roots and shoots of plants. Secondary hardening in shade houses. Shade houses are placed on the field. They reduce some amount of sunlight and temperature. Shade houses are provided with raised beds, proper drainage, micro irrigation, and fertigation arrangement for individual plants in polybags, and misting system for microclimate control. In shade houses, polybags are filled with soil and organic and inorganic fertilizers. Before filling the soil is also cleaned and fumigated by various fumigants. Then fumigated soil are used for filling the bags, having the proper amount of organic and inorganic fertilizers, which is required for the better growth of plantlets. The plants are coming from greenhouses in portraits are removed according to the height and planted into the plastic bottles in soil. Here, irrigation and fertigation is provided by the sprinklers, and in secondary hardening, that means in shade houses, the plants are placed around again 45 days and it is completely hardened into the atmospheric condition, and then plants are transported towards the actual site of plantation. The plantlets after acclimatization should be transported to the required place for plantation. This is the end product of the tissue culture laboratory. That means first selection of suckers to completely harden the tissue culture plants. The complete journey of plants required 7 to 9 months. Students, we learn the basic structure of tissue culture laboratories in first section. Second section, we learn how to prepare the tissue culture plants from explants, and the third stage of our presentation is advantages of bamboo tissue culture. What are the advantages of tissue culture of bamboo plants and are true to the type of mother plant under well management? Students, the plants we prepared from one explants after inoculation, we prepared thousands of plants from one explants, and all plants, prepared plants, are completely identical to the mother plants from we took the explants for inoculation. That means the 100% characters of mother plants are come into the each and every plants we prepared through tissue culture. Next benefit is pest and disease free seedlings, because after selection of explant, we transported each and every explant to the virus indexing laboratories. And in virus indexing laboratories, the disease free, virus free plants are only transported toward the tissue culture laboratory for actual inoculation or actual production of tissue culture plants. The complete disease-free, virus-free explants are used for the production of tissue culture plants. That means each and every plants we prepared through tissue culture are 100% pest, disease and virus free. Number 3, uniform growth and increased. Because each and every plants having the same age group, that means they are uniformly growth and 100% increased yield because we select the mother plants for taking explants. The selection of mother plant is the most important criteria. So naturally we prepared plants are also increases the yield. Early maturity of crop, maximum land used is possible in low land holding country like India. Around the year planting possible as seedlings are made available throughout the years. Students, bamboo plants having the seeds after 30 to 40 years. It is a very much long time for taking seeds. So tissue culture is the best way to prepare a number of plants for plantation, and in any month, any day, we prepared the tissue culture plants and we planted throughout there. There is no problem to plant the tissue culture plants. No stagger harvesting. New varieties can be introduced and multiplied in a short duration. We also prepared new varieties through the tissue culture or breeding techniques to conserve rare and endangered species. Those plants, bamboo plants, are going to be endangered or rare. We can prepare a number of plants through tissue culture and planted into various forests. So the rare and endangered plants can multiply it, and the number of plants of rare and endangered are increased. So we can conserve the rare and endangered species through tissue culture techniques. Thank you, students.